Good morning, students. Today we are discussing cell, the unit of life. We can expect around 4 to 6 questions from this chapter for our Neet examination. And we know that cell is the basic structural and functional unit of life. All living organisms are composed of cell, and cell is the basic structural and functional unit of life. In the single-celled organism like amoeba or bacteria, this is the cell, and the single cell has independent existence. Single cell has independent existence, and it can perform all the essential activities of a life like food capturing, digestion, excretion, reproduction. Everything is done by a single cell, and that's why they are called as single-celled organism or unicellular organism. But in the case of multicellular organism, there is the hierarchy, that is the cell grouped to form tissues grouped to form organ, and in the case of animals, the organ grouped to form organ system, and different organ system grouped to form the organism. So in a multicellular organism, there are different types of cells are present, and the cells grouped to form tissues grouped to form organ and various organ system, and also ultimately organism. But in the case of single-celled organism, the single cell which perform the independent existence is present in the single-celled organism, and the single cell that perform all the essential activities of life. And who discovered cell? The cell was first discovered by Robert Hooke in the 17th century, and it was a dead cell, and he discovered the cell from cork slices by using his crude microscope, and the cork plant is scientifically called as Quercus subur. The commercial cork is obtained from Quercus subur, and he taken the slices of cork plant and he observed as honeycomb-like structure. Actually, we know that in the case of cork plant, the suberin is highly deposited on the cell wall. He discovered only the cell wall, not the living cell, and he explained his discovery in his book called Micrographia. Micrographia is the book by Robert Hooke, and he used the term cellula, which means compartment. From that term, cell was originated. So the first cell, that is not a living cell, it is a dead cell. It was discovered by Robert Hooke, and later Anton Levin Hooke discovered a living cell, that is a bacterial cell. And in the 18th and also in the 19th century, so many scientists like Robert Brown, George Palade, Camillo Golgi, and numerous scientists, they discovered different cellular organs and their functions. And the next important topic, that is cell doctrine or cell theory. And what is cell theory? Cell theory was proposed by two scientists, Theodor Schwann and Matthias Schleiden. In 1838, Theodor Schwann, a British zoologist, and in 1839, Matthias Schleiden, a German botanist, conducted experiments and studies on plant and animal body, and they concluded that all organisms are composed of cells and cell products. And that's why we always say that the statement cell is the basic structural and functional unit of life. That comes from the first postulation of cell theory proposed by Theodor Schwann and Matthias Schleiden. All organisms are composed of cells and cell products. After 16 years, that is in 1855, the cell theory was modified by Rudolf Virchow, a German biologist. He conducted the cell division or the cell formation, and he introduced the statement hominis cellulae cellula. Hominis cellulae cellula, which means all new cells arises from pre-existing cell. We know that a new cell is originating only by the division of the previous cell. So this is the cell theory. Cell theory was proposed by Theodor Schwann and Matthias Leiden in the year 1838 and 1839. The years are very important, and the name of the scientist and also their branch of science, their country, everything is asked in the examination, and they propose the first statement. All organisms are composed of cells and cell products, and later in 1855 the cell theory was modified by Rudolf Virchow by adding the statement hominis cellula. Hominis cellula, which means all new cells arises from pre-existing cell. Only the pre-existing cell undergo division and form new cells. So this is about cell theory, and cell theory is also called as cell doctrine, and it is a very important part, and micrographia is an important one, and the term cellula was introduced by Robert Hooke, and it was first mentioned in the book Micrographia. Cellula, which means compartment. Cellula, which means compartment. He observed honeycomb-like structure, and Robert Hooke used cork slices. The scientific name of cork plant is Quercus tuber. And in the single-celled organism, the single cell has independent existence, and it performs all the essential functions of life. So this is the first part of the cell, the unit of life is the overview of cell. We know that there are two types of cells, prokaryotic cell and also eukaryotic cell. So now we are discussing the differences between prokaryotic and eukaryotic cell. What is prokaryotic cell? Prokaryotic cell, pro which means primitive or first, and karyo is nucleus. So they are primitive nucleated cell. And in the eukaryotic cell, U means true and karyo is nucleus. So they are true nucleated cell. And what is primitive nucleated cell? In a cell, in a bacterial cell, there is cell wall is present and a cell membrane is there, and in the cytoplasm the free genetic material, or the genetic material, the DNA, the circular DNA which is present free in cytoplasm, that is called as naked genome or naked DNA, and they are called as nucleoid. Oid, which means like that. Oid, O-I-D. That is, they are like nucleus, they are performing the function of nucleus, but it is not a true nucleus. And what is a true nucleus? That is, the nuclear membrane is present, that is, the genetic material is covered by a membrane, a nuclear membrane is present. So in the case of eukaryotic cell, there is a nuclear membrane, double layered nuclear membrane, and a fluid part that is called as nucleoplasm is present, and in the nucleoplasm the chromatin material or DNA is present, and also nucleolus is present. This is a true nucleus, and a cell with a true nucleus or eukaryote, they are called as eukaryotic organism. The eukaryotic organism, nuclear membrane is present, and the genetic material they coiled and condenses to form specific chromosome, and nucleolus is present, and this is the structure of a true nucleus or eukaryote. And what is another difference? Membrane bound organelles such as mitochondria, chloroplast, endoplasmic reticulum, Golgi body, etc. are totally absent in the case of prokaryotic cell. Is it clear? That is, in the case of prokaryotic cell, membrane bound organelles are totally absent, but in the case of eukaryotic cell, membrane bound organelles, all different types of membrane bound organelles or the cytoplasmic complexities are present in the case of eukaryotic cell, such as mitochondria, chloroplast, endoplasmic reticulum, Golgi body are present in the case of eukaryotic cell. So what are the differences? First one is, it is a primitive nucleated cell. Pro is first or primitive, and they have no nuclear membrane. The DNA is naked, a circular DNA, a coiled DNA which is present in the free in the cytoplasm, and that is called as nucleoid. But in the case of eukaryotic cell, it is a true nucleated cell with a nuclear membrane and chromatin material, and nucleolus is present, a true nucleus is present. But in the case of prokaryotic cell, membrane bound organelles are totally absent. Yes. And in the eukaryotic cell, membrane bound organelles are present. And in the case of ribosome is common in both the prokaryotic and eukaryotic cell, and ribosome is not a membrane bound structure. Okay, and in the ribosome, a 70S type of ribosome is present in the case of prokaryotic cell, and 80S ribosome is present in the case of eukaryotic cell. Ribosome, 70S ribosome is present in the case of prokaryotic cell, and 80S ribosome is present in the case of eukaryotic cell. And in the case of flagella is a locomotory organ present in many of the prokaryotic, especially protistants, or in the case of protozoans, and also higher organisms also have flagella. In our body also have flagella, and this flagella, in the eukaryotic flagella, there is a 9+2 arrangement of microtubule. 9+2 arrangement of microtubules present in the eukaryotic cell, but in the case of prokaryotic cell, no specific arrangement of microtubule is present. There is no specific orientation or arrangement of microtubule is present in the case of prokaryotic cell. And we know that prokaryotic cell, which was represented by kingdom Monera only. We know kingdom Monera is an exclusive kingdom for prokaryotic organism. And eukaryotic cells are represented by the kingdom Protista, fungi, plantae, animalia. All other kingdoms of the 5 kingdom classification are represented by eukaryotic cell. But in the case of prokaryotic and eukaryotic cell, when we are comparing the prokaryotic and eukaryotic cell, only one structure is common and similar. Chemically similar and common in both the prokaryotic and eukaryotic cell. That is only one structure, that is cell membrane or plasma membrane. Plasma membrane. Normally there is a question like which of the following is common and similar, or structurally similar, or chemically similar in both the prokaryotic and eukaryotic cell. Either they will be use the term prokaryotic or eukaryotic cell, or they will be give example bacteria and euglena, or bacteria or amoeba. In both the prokaryotic and eukaryotic cell, the common structure is not, it is the common and chemically similar structure is not a ribosome or cell wall or not cytoplasm. Because only one structure that is common and similar in both the prokaryotic and eukaryotic cell, that is cell membrane. So cell membrane is chemically similar of eukaryotic cell. The cell membrane of prokaryotic cell is similar to that of the eukaryotic cell. So that is a very important point. So cell membrane is common and similar, chemically similar in both prokaryotic and eukaryotic cell. So this is the comparison between two cells, prokaryotic and eukaryotic cell. And in the case of eukaryotic cell, there are membrane bound organelles are present. But in the case of animal cell, there are ribosomes are present. Ribosome is common in both the prokaryotic and eukaryotic cell. But in the animal cell, there is a non-membrane bound cell organelle called as centriole. Centriole is a non-membrane bound structure that is present only in the case of animal cell, not discovered in the case of higher plants, and they are non-membrane bounded structure. It has no membrane, and it is acting as the basal body for cilia and flagella, the origination of cilia and flagella, and also the centriole is involved in the formation of spindle fiber during cell division, mitosis and meiosis. So centriole is a non-membrane bound structure present only in the animal cell, not in plant cell, and it is involved in the cell division for the formation of spindle fiber, and it acts as the basal body for the cilia and flagella. So this is the comparison between prokaryotic and eukaryotic cell. So next is there are certain cells that is described in in our textbook, that is some cells, only some cells and their shape and an overall idea of the size we are discussing. So the shape of the cell and example is given as per NCERT, and the first shape, that is round biconcave. Round biconcave shape was represented by RBC of human body, and the second is round oval shape was represented by mesophyll cells of plants, and the third is long branched cell was represented by neuron or nerve cell, and the fourth one is long narrow cell was represented by columnar epithelial cell, and the fifth one, long narrow amoeboid cell. Amoeboid is by WBC. So round biconcave was represented by RBC, round oval by mesophyll cell, long branched cell by neuron, long narrow cell by columnar epithelium, and amoeboid shape by WBC, and the sixth one, elongated cell by xylem tracheids. So the shape of the cell is different. The shape of the cell and the shape and structure of the cell is designed to perform which function it is performing. The shape and structure of the cell is designed to perform which function, that we can see here. That is, in the case of round biconcave cell, it is RBC which carry oxygen and which transport gaseous molecule throughout the body. So it is carrying maximum oxygen, so it has a shape of round biconcave. But in the case of WBC, it is amoeboid because it has to undertake phagocytosis. And in the neuron, it is long branch to conduct nerve impulses very fastly. So this shape is given as per NCERT. So it is very important. They may be given for matching type or wrong pair or correct pair. So the 6 shapes and 6 sizes also mentioned in the NCERT, and the sizes of the cell and example. First one is bacteria. Bacteria is with 1 to 5 micrometer in size. Bacteria has a range of 1 to 5 micrometer, but mycoplasma has 0.1 to 0.3 micrometer. We know mycoplasma is structurally similar to bacteria. Both are belonging to Monera, but they do not have cell wall. So their cell is very small. And in the case of PPLO, pleuropneumonia-like organism, it's the smallest living cell, and it has a 0.1 micrometer. 0.1 micrometer. And the next shape, next size, that is a typical eukaryotic cell, or that is in the case of RBC, 7 micrometer. And in the case of typical eukaryotic cell, that is about 10 to 20 micrometer. And in the case of virus, it is about 0.02 to 0.2 micrometer. And we know that it is smaller than mycoplasma, but we do not consider virus is as a smallest organism because they have no cellular organization, and virus which do not follow cell theory because it is not composed of cell, that we discussed in the chapter biological classification. So these are the different sizes: bacteria, mycoplasma, PPLO, RBC, typical eukaryotic cell, and also in the virus. So the shapes and the sizes are different. So the shape and size is different in different organisms. So next we are moving to the structure of a prokaryotic cell and structure of a eukaryotic cell. Next is the shape of prokaryotic cell. Prokaryotic cell was represented by kingdom Monera, and in the kingdom Monera the sole member is bacteria. On the basis of shape of bacteria, bacteria can be classified into 4 different groups: Bacillus, Coccus, Vibrio, and Spirillum, that we have already studied in the chapter biological classification. Bacillus is rod-shaped bacteria, Coccus is round-shaped bacteria, Vibrio is comma-shaped bacteria, and Spirillum is spiral-shaped bacteria. So the prokaryotic cell, it is represented by only kingdom Monera, and the sole member is bacteria, and it also includes archaebacteria and mycoplasma and also cyanobacteria. And what is the structure of a prokaryotic cell or a typical bacterial cell? In a bacteria, there is a genetical material, and the genetical material is naked, and the naked genetic material, that is called as nucleoid. Naked DNA is called as nucleoid, and that is free in cytoplasm. It is not bounded by any membrane. It is not bounded by any membrane. And in addition to, in the case of prokaryotic cell, in the case of bacteria, many bacteria, they have a extra chromosomal circular DNA. Extra chromosomal circular DNA, and that extra chromosomal circular DNA is called as plasmid. So the genetic material of the bacteria is nucleoid or naked DNA, and there is a extra chromosomal circular DNA present in prokaryotic cell, and that is called as plasmid. And we have studied in the chapter biotechnology, we are using plasmids for rDNA technology or recombinant DNA technology or genetic engineering as cloning vehicle. And plasmids which provide antibiotic resistance, and also they provide specific genetical or specific sexual behavior to the bacteria. So plasmids are the extra chromosomal circular DNA. Plasmids are the extra chromosomal circular DNA present in prokaryotic cell or in the bacterial cell. And the cell envelope of bacteria, which is chemically complex. The cell envelope of bacteria, which is chemically complex, and there is an outermost layer, and that is called as glycocalyx. The outermost layer of cell envelope of bacteria, it is called as glycocalyx. The cell envelope of bacteria is chemically complex, and it is composed of 3 distinct layers, but they are compactly arranged one above another. The outermost layer is glycocalyx. When the glycocalyx is very tough and rough, it is called as capsule. When we use the term capsulated bacteria, that says that it has capsule and it has very hard glycocalyx. And when it is loose sheath, it is called as slime layer. So outermost layer is glycocalyx, and the middle layer is cell wall, and the cell wall is composed of peptidoglycan. Peptidoglycan, that we have already discussed in the previous chapter. Peptidoglycan composed of amino acid and polysaccharide. Murine or muramic acids are the major chemical component of the cell wall of bacteria. And in the case of bacteria, the third innermost layer, that is cell membrane. Cell membrane of bacteria is very important because it is structurally and chemically similar with eukaryotic cell. And in the case of cell membrane of prokaryotic cell or bacterial cell, there is a specialized structure. There are certain invagination present on the cell membrane, and that is called as mesosome. Mesosomes are very important for the bacterial cell, and mostly the questions are asked in the mesosomal part, because mesosomes, which are 3 different types, one may be vesicular, this is vesicular, or tubular, or lamellar, and the mesosomes are involved in cell division, DNA replication and secretion, and also it is involved in the respiratory process, because if it is a membrane bounded structure or it is a invagination of the plasma membrane, if that contain the enzymes that is required for respiratory process, that will undertake respiration, or that is involved in the different enzymes, different chemicals are present in the specified mesosome, and these mesosome, they are performing their own function. So mesosomes are very important in the case of prokaryotic cell. They are performing the various metabolic activities like respiratory process or the secretory process, and also DNA replication, cell division, cell wall formation, and even certain chromatophores are present in the photosynthetic bacteria or cyanobacteria, and they are filled with pigment, photosynthetic pigment, and undertake photosynthesis. So chromatophores. So mesosomes are the invagination of the plasma membrane, and it may be three different shapes. One may be vesicular or tubular or lamellar, and they are performing various metabolic activities. And evolutionists, cytoevolutionists, they consider the mesosomes are forerunner of cell organelle, because from this structure cell organelle may be originated by joining the cell membrane. Okay, so the cell membrane may be originated from mesosome. Mesosomes are the primitive structure of membrane bound cell organelle. So the cell wall or envelope, it has three different layers: glycocalyx, cell wall, and cell membrane. Cell membrane has two important properties. One is it is structurally and chemically similar to eukaryotic cell, and the second is it has mesosome. Mesosomes are carrying out various metabolic activity. And the organelle ribosome, we know that ribosomes are the site of protein synthesis, and majority of ribosomes of prokaryotic cell or bacterial cell seen attached with plasma membrane. And 70S ribosomes are present in the prokaryotic cell. 70S ribosomes are present in the prokaryotic cell. And next is inclusion bodies. What is inclusion body? In the prokaryotic cell, the non-membrane bound material like the reserve food material may be present in the cytoplasm without any membrane. And such reserve food material without any membrane, such as they are called as inclusion body. And inclusion body that include phosphate granule, cyanophysian granule, and glycogen granule. These are the inclusion bodies present in prokaryotic cell. And the prokaryotic cell, there are certain cellular outgrowth like flagella, pilus, and fimbriae are present. Flagella is the longest structure and which is helping in the movement of the bacteria, and it has three parts. One is hook, basal body, hook, and filament. Pilus is finger-like outgrowth. It is composed of pilin protein, and pilus which provide certain sexual behavior to the bacterial cell. Pilus is, which are elongated finger-like outgrowth which is present on the bacterial cell, and it is composed of a special pilin protein. And fimbriae are bristle-like structure, and it helps to attach with the host cell and also in the rocks. So fimbriae are bristle-like structure which is present throughout the surface of the bacterial cell, and it helps to attach with the host cell. So this is the overview, or this is the basic structure of a bacterial cell: nucleoid, plasmid, cell envelope, and mesosomes. What is the characteristic features of glycocalyx, cell wall, cell membrane, and the different types of mesosome: vesicular, tubular, and lamellar. Characteristic features of ribosome and inclusion body, and membranous outgrowths like flagella, pilus, and fimbriae. Okay, I think you understand about the overview of the bacterial or prokaryotic cell. Next, we are moving to the eukaryotic cell. We know eukaryotic cell have true nucleus, membrane bound cell organelles like mitochondria, Golgi body, endoplasmic reticulum, chloroplast, etc., and 80S ribosome, and plasma membrane, nucleus, everything is present. So the complex cellular structure is present in the case of eukaryotic cell. And eukaryotic cell was represented by protista, fungi, plant and animal kingdom. And we are going to discuss the structure of a eukaryotic cell. And a eukaryotic cell, they have plasma membrane. Plasma membrane is chemically similar to that of prokaryotic cell. And the chemical analysis of plasma membrane that shows that it is composed of phospholipid and protein. Phospholipid and protein. The chemical analysis of plasma membrane that shows that phospholipid and protein, and the phospholipid which form the bilayer, and the phospholipid which has two parts, a hydrophilic head and a hydrophobic tail. And there are two types of protein, intrinsic protein and extrinsic protein. And the widely accepted model of plasma membrane was proposed by S.J. Singer and J.L. Nicholson, and that is called as fluid mosaic model. Before, there are unit membrane concept and Daniel E. Davison sandwich model. And the widely accepted model to explain the structure and function of plasma membrane was given by S.J. Singer and J.L. Nicholson in 1972, and it was fluid mosaic model. And according to the chemical composition conducted by the studies by the Singer and Nicholson, it is composed of mainly phospholipid and protein. And this is the structure of the fluid mosaic model. As per the diagram, we can see that there are phospholipid bilayer. A phospholipid bilayer has hydrophilic head and a hydrophobic tail. And there are two types of protein. One is intrinsic protein that you can see in the diagram, and the extrinsic protein, and it is present outside the membrane. And the intrinsic protein are mainly act as the carrier protein during the facilitated diffusion and also active transport. And the widely accepted model, fluid mosaic model, it has certain benefits, that is, it has certain merits, because it can explain all the functions performed by plasma membrane, like the movement of molecule, membrane transport, and also cell junction and cellular communication, cell recognition. Everything can be explained by the model, the fluid mosaic model. And according to the chemical analysis of the plasma membrane of RBC, it is composed of, the plasma membrane of RBC that composed of 56% of protein and 40% of lipid. 56% of protein and 40% of lipid is present in the plasma membrane of RBC. And what is the fluidity of plasma membrane? Fluidity of plasma, according to the fluid mosaic model, plasma membrane is a quasi-fluid membrane. Quasi-fluid membrane, and the phospholipid layer which provide fluidity. Fluidity is the ability of protein molecule. Fluidity is the ability of protein molecule to undergo overall lateral movement in the lipid bilayer. So the fluidity is provided by lipid bilayer. Because lipids are not compactly arranged, it is semi-fluid or quasi-fluid in nature. So the lipid protein can undergo overall movement, and that is helping in the carrier transport, like facilitated diffusion and also active transport. So we know that the structure, chemical composition of plasma membrane is common in both prokaryotic and eukaryotic cell. So this is the structure of plasma membrane according to the fluid mosaic model proposed by S.J. Singer and J.L. Nicholson in the year 1972. Next, we are moving to different cellular organelles. We know cytoplasm is the fluid matrix of the cell, and cytoplasm is a major arena of various chemical reactions of the cell, and the chemical reaction make the cell alive. And the cytoplasm, which have different cytoplasmic organelles like mitochondria, Golgi body, endoplasmic reticulum, lysosome, chloroplast, etc., they are performing different functions, and these different metabolic functions of the cellular organelles and cytoplasmic organelles make the cell alive. So we are moving to study the different cellular organelles, such as endomembrane system. Endomembrane system is a membranous system. They are functioning coordinatingly, and it include 4 different cell organelles like endoplasmic reticulum, Golgi body, lysosome and vacuole. So we are going to study endomembrane system. Endomembrane system include 4 cell organelles. One is endoplasmic reticulum, second is Golgi body, third is lysosome, and fourth one is vacuole. They are called as a part of endomembrane system because their functions are in a coordinated manner. And endoplasmic reticulum, which is seen near with the nucleus, so this is the nucleus. See, near with the nucleus there is tubular part, and this tubular part is made of double layered membrane, and it is called as endoplasmic reticulum, and which form a network or skeletal work inside the cell, and which act as the transport of material throughout the cell. And there are 2 types of endoplasmic reticulum. The endoplasmic reticulum seen near with the nucleus are the presence of ribosome, so their surface is rough, and they are called as rough endoplasmic reticulum, and due to the presence of ribosome they are the site of protein synthesis. So rough endoplasmic reticulum is a site of protein synthesis. But certain endoplasmic reticulum distant from the nuclear region, which are devoid of ribosome in their surface, so their surface is smooth, so they are called as smooth endoplasmic reticulum, and they involved in the synthesis of lipid. So in our endocrine cells, or the especially the steroid hormone producing cells, they have large number of smooth endoplasmic reticulum. So in the animals or in the human body, the steroid hormones are mainly synthesized on the smooth endoplasmic reticulum. So endoplasmic reticulum is of 2 types. One is rough endoplasmic reticulum, and the second is smooth endoplasmic reticulum. And second part of the endomembrane system is Golgi body. It was discovered by Camillo Golgi, and it is very close to endoplasmic reticulum, and they modify the biomolecule that is synthesized by rough endoplasmic reticulum or smooth endoplasmic reticulum, like protein or lipid, into glycoprotein and glycolipids, or they become activated or modified into the active form by the Golgi body. So they are involved in the packaging and secretion of the material. So first is endoplasmic reticulum, and to the endoplasmic reticulum very close structure, that is Camillo Golgi discovered Golgi body or Golgi apparatus, and the Golgi apparatus is involved in the packaging and secretion of biomolecule that is synthesized by the endoplasmic reticulum. And the third is lysosome. Lysosome was discovered by D. Du in 1935. And lysosomes are commonly called as suicidal bags of the cell because they are filled with hydrolytic enzymes like lipase, carbohydrases, proteases and nucleases, because they are containing the enzyme that degrade a cellular component. So it is active in the case of the immune cells like phagocytic cells or WBC cells. When it captures the foreign particle, they release the hydrolytic enzyme and that degrade the foreign particle. And these hydrolytic enzymes are optimally active at acidic pH. They are optimally active at acidic pH. So lysosome is formed by the packaging activity of Golgi body. Lysosomes are formed by the packaging activity of Golgi body. And the last is vacuole. We know vacuole is very prominent in the case of plant cell. About 90% of total volume of the plant cell is occupied by vacuole, and vacuole is made of unit membrane or single layered membrane, and that is called as tonoplast. It is single layered membrane, and that facilitate the movement of a number of ions against concentration gradient, because vacuole in the plant cell that store numerous waste material and excretory material and cell sap and the material not useful for cell. So it is in the case of plant cell the vacuole which store the waste material. And the vacuoles like a contractile vacuole or food vacuole is present in the case of protistants or protozoans, because for the food capturing and also excretion they are involved. So vacuole is prominent in the case of eukaryotic cell. The prominent vacuole is present in the plant cell, and about 90% of plant cell is occupied by vacuole. and the vacuole is covered with a single layered membrane, and that is called as tonoplast. So this is the part of endomembrane system: endoplasmic reticulum, Golgi body, lysosome, and vacuole. Next we are moving to discuss the mitochondria and chloroplast. Mitochondria and chloroplast are two semi-autonomous cell organelle. So we have to discuss mitochondria and chloroplast. Mitochondria is common in both plant and animal cell, but the chloroplast is present only in the photosynthetic organism. And both are called as semi-autonomous cell organelle. And what is the shape of mitochondria? We know mitochondria is a site of respiration and it is a site of ATP synthesis. And mitochondria is commonly called as powerhouse of the cell, because it is a site of cellular aerobic respiration, Krebs cycle and electron transport chain, and it is a site of ATP synthesis. And mitochondria has a length of 1 to 4.1 micrometer and a diameter of 0.2 to 1.0 and an average of 0.5 micrometer diameter. And mitochondria has outer membrane and inner membrane, and inner membrane has invaginations, and it is called as cristae that increases the surface area of mitochondria. And the cristae that contain F0 F1 particle, we know we have already studied about F0 F1 particle or oxysome in the case of respiration or aerobic respiration. This is F0 F1 particle, and here the ATPase or ATP synthetase enzyme was present, and it is a site of ATP synthesis. The F1 particle is a site of ATP synthesis. And the mitochondrial matrix that contain various enzymes for the Krebs cycle, and it is a site of Krebs cycle, and mitochondrial inner membrane is a site of ETS or electron transport chain. And mitochondria contain a strand of DNA and some RNA and also ribosome. But the ribosome present in the mitochondria is not 80S ribosome, it is 70S ribosome. Due to the presence of DNA and ribosome, mitochondria has synthesized their own biomolecule to a certain extent, and that's why mitochondria is called as semi-autonomous cell organelle. Mitochondria is the powerhouse of the cell. Next is chloroplast. We know chloroplast is a kitchen of the plants, and commonly it is called as plastid. And plastids can be divided into three types. One is chromoplast, second is leucoplast, and third is chloroplast. Chromoplast, leucoplast and chloroplast. Chromoplasts are colored other than green color, yellow, orange, red, etc. due to the presence of xanthophyll and carotene. Due to the presence of the pigment xanthophyll or carotene, and they provide a particular color to plant parts like floral parts or certain leaves. They are providing different varied color. So the pigment, the cellular organ and the plastid which is present in the colored part is chromoplast. And leucoplast, which is involved in the storage of food material, and it can be divided into three basic types. One is alluroplast, which store protein. Alluroplast which store protein. And the next is eleoplast, which store fat molecule. And the third is amyloplast, which store starch molecule. So alluroplast which store protein, eleoplast which store fat molecule, and amyloplast which is store starch, and the leucoplast is the storage cell organelle or plastid which do not have any color, no color. And chloroplast is green in color. Chloroplast is a photosynthetic structure, and the chloroplast has a length of 5 to 10 micrometer. That is, chloroplast is bigger than mitochondria, and it has a diameter of 2 to 4 micrometer. And chloroplast has the grana thylakoids arranged one above another from grana. Grana contain chlorophyll, and the fluid matrix, it is called as stroma. Stroma is a site of the C3 cycle or carbon dioxide reduction and or a dark reaction, and it also contains a strand of DNA and a certain ribosome is also 70S ribosome. So in an eukaryotic cell, both the mitochondria and chloroplast has 70S ribosome. 70S ribosome, and they are called as semi-autonomous cell organelle. So the structure of mitochondria and structure of chloroplast are designed to perform their functions like ATP synthesis or cellular respiration and also nucleus. Nucleus is the controlling unit or center of the cell. And nucleus was discovered by Robert Brown in 1831. A nucleus has an outer and inner membrane, and in between the membrane there is a perinuclear space of 10 to 50 nanometer. 10 to 50 nanometer is the perinuclear space. There is an outer membrane and there is an inner membrane, and in between outer and inner membrane there is a space, and that is called as perinuclear space. And perinuclear space is with 10 to 50 nanometer. Nucleus was discovered by Robert Brown in 1831. And there is a small, the outer and inner membrane is not continuous. It join together and form a small minute opening on the nuclear membrane, and that the opening present in the nuclear membrane, that is called as nuclear pore. Nuclear pore. It is a path for both the movement of inside the nucleus and from outside the nucleus. The RNA and RNA will be moved out of the nuclear membrane, and the synthesized protein may be moved inside the nucleus. So this is the bi-directional transporting region that is called as nuclear pore. So nuclear pore which will be transport RNA to the outer side and protein into the inner side. And the fluid matrix of the nucleus is called as nucleoplasm. And nucleoplasm that contain two parts, one is chromatin and nucleolus. Chromatin is the highly coiled, tangled structure of the genetic material, and the term chromatin was discovered by Fleming. The term chromatin was introduced by Fleming. The term chromatin was introduced by Fleming, and the chromatin undergo condensation during the cell division and form specific chromosome, mitotic chromosome. And a mitotic chromosome is composed of two chromatids, sister chromatid, held together by a centromere. So the chromatin during the cellular division, they condenses to form specified chromosome, and there is a centromere and two chromatids. These are two chromatids, sister chromatids are there. In the centromere, there is a proteinaceous discoid region is present, and it is a site of attachment for the spindle fiber or microtubule during the cell division. And this region, this site of attachment of microtubule, that is called as kinetochore. That structure is called as kinetochore. So each chromosome has two kinetochore. One is on this side and another is on that side. So each kinetochore is a site of attachment of spindle fiber from either side. And if a cell which carry 16 chromosome, that has 16 into 2, 32 kinetochore will be present inside the cell. So the number of kinetochore is very important. Each chromosome has two kinetochore. Kinetochore is a discoid proteinaceous structure, and it is a region for attachment of microtubule. So the chromatin is a coiled region, and it is a DNA or the nuclear material. And the chromatin, the term was introduced by Fleming, and the chromatin material condenses to form specified chromosome during the cell division. And inside the nucleoplasm, there is a condensed non-membrane bound structure present, and it is called as nucleolus. And nucleolus was discovered by Fontana. And nucleolus is a site of, nucleolus was discovered by Fontana, and nucleolus is a site of rRNA synthesis. rRNA synthesis. So the nucleolus is involved in the rRNA synthesis. Nucleolus is involved in the rRNA synthesis. That's why when a cell is actively involved in protein synthesis, they have large nucleolus or more than one nucleolus per cell. When a cell is actively involved in protein synthesis, they have large nucleolus or more than one nucleolus may be present, because the nucleolus is a site of rRNA. We know that rRNA is a structural component of ribosome. So the ribosomal assembly or ribosomal formation is taken place in the nucleolus. So nucleolus is a non-membrane bound structure and it is involved in the rRNA synthesis or ribosomal synthesis. So what is a eukaryotic nucleus? Eukaryotic nucleus was discovered by Robert Brown in 1831, and it has a two membrane, outer and inner membrane. In between outer and inner membrane there is a space called perinuclear space, and the perinuclear space is about 10 to 50 nanometer, and nuclear pore is a bidirectional path for RNA and protein, and the nucleoplasm is a fluid matrix of the nucleus, and it has chromatin and also nucleolus. The chromatin is a genetic material. Chromatin, the term was introduced by Fleming, and the chromatin undergo condensation during the cell division, forms specified chromosome with mitotic chromosome, two sister chromatid held together by a centromere, and centromere has specified proteinaceous structure for the attachment of the microtubule, and that is called as kinetochore. So each chromosome has two kinetochore. And nucleolus is a non-membrane bound condensed region, and it was discovered by Fontana, and nucleolus is a site of rRNA synthesis and ribosomal assembly. So that's about the chapter Cell, the Unit of Life, and it is a very important chapter for the NEET examination, and it is a basic chapter for biomolecule and the cell, the unit of cell cycle, and also the chapter like reproduction, and also the chapter like molecular base of inertance, because cell is the basic unit of life. Study well and do all the question that is given in our material and also the previous question.
Bright Education Center, Medical and Engineering Entrance Coaching Center, Hilal area, Doha, Qatar. For further enquiries, contact 306 00679.